Hello, everyone. I am Shuyu. I am a Ph.D. student at Penn State University. This is my teammate, Pletcher. She is an undergrad senior at Michigan State University. Today, we are here very excited to share with you guys a project we worked on during our summer internship through the NASA Develop program. And in this project, we utilized remote sensing, machine learning, and Earth data observations to improve the detection systems of harmful algal blooms in the central Texas in Highland Lake chain.
Hi, everyone. So as you can see in this map, coastal regions worldwide have been experiencing increased incidences of algal events and hypoxic zones worldwide. So some of the effects of this are altered composition in water columns, as well as disruptions in nutrient cycling, among many other things. So because of this is currently one of the greatest threats to coastal ecosystems worldwide. This is also affected in inland regions as well, as so greatly demonstrated by Lake Erie in the Midwest. But for our purposes, we are going to be focusing on a Texas lake chain in central Texas. Next slide. So, as we know, algae isn't inherently harmful, but in excess, it can cause a lot of problems in ecosystems. Not only is this detrimental to the ecosystem, as we just talked about, but it's also extremely detrimental to local economies as well as public health. So in Austin specifically, Lady Bird Lake and Lake Travis are very metropolitan areas, and they are affected by closure of public areas and beaches, which results in a huge loss of revenue for the area. More importantly, though, this algae can result in the production of harmful neurotoxins. So specifically, Austin started experiencing the effects of this in 2019. When several Austin residents reported their dogs were dying after consuming this algae. So this is a problem that nobody wants to see. We all love our dogs, so that's why we care. Next slide. So like I said, our area of interest is a lake chain in Texas. It is the Highland Lake chain. This is composed of a series of dammed reservoirs within the Highland Lakes and the lower Colorado River. Over here to the left, we can see Lake Austin and Lady Bird Lake. These are our two most interesting lakes because they are geographically extremely close to downtown Austin. Specifically, they have high levels of tourism year-round. Our study period was constrained by the launch of our utilized satellites. So we began in 2013 with the launch of Landsat 8, and our data is being updated to the present period. Next slide. There should be one more. Go back. Yeah. So in order to achieve these goals, we used two satellites. We utilized the jointly operated NASA USGS Landsat 8 satellite. It has a longer revisit time, but it has pretty good spatial resolution. On the other hand, we also utilized ESA's Sentinel-2 satellite, which has higher temporal resolution thanks to multiple sensors. And it also has more sensor capabilities and therefore can detect more spectral bands, which is helpful for our detection of proxies.
So in this project, our objective is to provide with our end users a user-friendly toolkit to help them better understand and monitor the algae conditions in our study area. So in this toolkit, environmental proxies retrieved from remote sensing products, such as chlorophyll A concentration, cyanobacteria detection, turbidity, and water surface temperature were all utilized in this toolkit. And technically speaking, the cyanobacteria detection was retrieved from the broad wavelength algae index, which is a modified version of cyanobacteria index. And this BWAI can help us capture both So the cyanobacteria in both the subsurface water column and also on floating mats, because this algorithm was designed to capture the spectrum peak at both grain bands and also near infrared bands. And then for stability, it was retrieved from a well-established normalized difference stability index, which can help us capture the spectral peak at the right wavelength. And for the water surface temperature, it is retrieved from harnessing the Landsat 8 thermal infrared band and also an inverting format of the Planck's law. And as for chlorophyll A concentration, it is retrieved from the a novel pre-trained machine learning model named Mixture Density Neural Network. And recently paper published by our NASA scientist, by Brendan Smith and Dr. Nima Polovin suggested this method has outperformed most of the state-of-art benchmarks. And standard neural network directly model the chlorophyll A concentration, while the mixture density neural network, it models the chlorophyll A concentration with a probability distribution through melting multiple Gaussian distributions over the outputs. And the mixture density neural network utilizes multiple bands in the visible bands and also near infrared bands. And it should be noticed that in this project, it is the first time that this machine learning model will be utilized in human-made lakes in reservoirs. Next slide, please. So after we get our data worked and also validated, we also created this user-friendly graphical tool or GUI. We call it Lake Algal Monitoring Dashboard to help our end users to visualize, download, and retrieve the information as needed. So the picture here shows how we conceptualize our GUI and how it looks like. So you can see we utilized interactive maps that allows our end users to toggle between layers and also zoom in and zoom out. We also employed multiple information widgets, which will show the metadata, statistics, and dates of the imagery on the map. At the same time, we also have a drop-down menu, which allows our end user to get the time series chart for the chlorophyll A or other variables like time series chart, and they can even download it. So all these components will help our end users to get their information, and it's very user-friendly. Next slide, please. So in the next couple slides, I will show the application of chlorophyll A concentration to help us better understand the algae conditions in this study area. So the top 6 figures here shows the Landsat 8 machine learning model retrieved chlorophyll A concentration in summers from 2015 to 2020 in the Highland Lake Chang. So basically, the deeper the color is, the higher the chlorophyll A concentration. And according to the Public Health and Water Quality Guidelines published by EPA in 2019, we can see these different levels of chlorophyll A concentration, cyanobacteria cells, and cyanotoxins level. So it's an effort to try to link between the response symptoms to the different level of exposure to the cyanobacteria. For example, the very high density of cyanobacteria cells corresponding to more than 50 microgram per liter of chlorophyll A concentration can be linked to very high risk to exposure to the macrocystins, one kind of cyanotoxins. Next slide, please. So beyond monitoring the near real-time algae conditions, we also try to understand the historical trend of algae conditions in both interannual and seasonal scales. So the figure here shows the time series chart of monthly chlorophyll A concentration for each lakes, from upstream to downstream. And basically, you can see the dot here shows the median value of chlorophyll A concentration for each month, and with one standard deviation of uncertainty. I think this plot actually shows us a lot of interesting pattern. So for example, you can see the chlorophyll A concentration in upstream lake is relatively higher than downstream lake. And also, if we take a close look at the Lake Travis, you can see our products capture the specs in 2016 and 2017, which corresponds to the severe drought, reported severe drought in 2016 and 2017. And if we take a look at this small spec in 2019, which corresponds to the reported harmful algae events in the Lake Travis. And as for seasonal variability, you can see here our data shows all the lake shows pretty significant seasonality of chlorophyll A concentration, and it peaks around the summer to mid-autumn, which overlaps with the seasonality of water surface temperature in this study area. And more interestingly, you can see the peaks delayed from upstream to downstream. So this is an ongoing project, and I can definitely see potentials from many other ways.
So the last product that we wanted to provide our end users with was that of Algal Hotspots. So that is where concentrations of chlorophyll A are high enough to be labeled high risk. So on this map, we can see that in the red regions. So one of the things we observed was this happening around the margins of lakes, which is something that we can expect. But we also detected it in and around man-made dams in the region, which is something that's interesting. So these types of observations are valuable to local policymakers because they can help plan future sampling efforts, mitigation strategies, and it can help us to monitor areas that we know are high risk. So the goal of this project was to So the goal of this project was to provide our end users, who were the Lower Colorado River Authority and the Department of Watershed Protection for the City of Austin, with tools in order to further understand what's happening in their water systems and in their community. So it's this combination of machine learning and remote sensing that allows for future problem solving, and that's something that's really important, and this will help in future efforts for risk communication as well as mitigation strategies in the city of Austin. At this time, we would like to thank you all for listening, and we would especially like to thank everyone on this list, each of whom were instrumental to the completion of this project. We'd like to highlight Katie, Mark, and Emma, who were our 3 team members on this project, as well as our NASA Develop Arizona node leader, Ryan Hammock. So thank you so much, everyone.